Hello and welcome everyone. In this podcast we're going to talk about mutations, and more specifically at the gene level. In the last several podcasts we were talking about mutations at the chromosomal level where we altered the whole chromosome or large parts of the chromosome. Now we're going to talk about mutations strictly at that one gene level. And I think it's important to recognize that gene mutations change the DNA sequence. With the chromosomal mutations, that would alter the expression of certain genes. It could remove certain genes. It could duplicate the copies of them. When we talked about epigenetics, that also is involving changing the expression of the genes. But now we're talking about specific mutations that actually change the sequence of the gene. And when we have a single mutation, a single nucleotide that's changed, we call these point mutations. And we can think of these as being base substitutions, or insertions and deletions. Now this is different than the chromosomal deletions or duplications that we talked about before, because with the chromosome we're talking about large segments of it. Here we're talking about single nucleotides being inserted or removed, or sometimes we might talk about 2 or 3 being added or removed, but we're talking at a much smaller scale here. And we're going to first talk about some examples of base substitutions. So let me go ahead and erase some on this board here. Okay, again we're talking about base substitutions. And we can break all base substitutions into one of two categories. Either transitions, and a transition is when you go from a pyrimidine to another pyrimidine, or a purine to another purine. The other type of base substitution is a transversion. And this is going from a pyrimidine to a purine, or vice versa, a purine to a pyrimidine. So that's what the base substitutions are. But now let's talk about the effect of these base substitutions. And so we're going to call these the effects of a mutation. And there are 5 that we're going to talk about. The first is a silent mutation. A silent mutation we can begin with by saying it has no effect. Alright, so let's go ahead and look at this silent mutation, and let's look at the DNA. And the DNA is GCT, and I've drawn it from using the template strand because that's how it's going to be read during transcription. As it's transcribed, we're going to make this following RNA, 5' CGA. And if you look at our code, and we'll put one over here real quick, you can see that a CGA is over here. CGA is arginine. So this is going to make an arginine. Now if we have a silent mutation, so we'll draw a line here and say mutation on it. Again, it's going to be a base substitution. Our new strand is going to be 3' to 5'. And so we're going to go, we'll keep that G, and we'll change that to C, and we'll make this a G. Now, again, this is the part that's been mutated. We've changed it from a T to a G. It gets transcribed now, and 5' to 3', it's going to be C, G, C. And if we put up our genetic code up here, let's put it up here so we can see it. We went from a CGA, which was here, CGA, gives us arginine. a CGC, CGC also gives us an arginine. So we call this a silent mutation because it does change the DNA. It does change this by making a substitution from a T to a G, but the end result is we still get an arginine. Now I'm going to just erase a part of this because we can use this same scenario here to look at another class of mutation. Okay, so let's talk about now this second category, and that's called a missense mutation. It's still a base substitution, but in this case we will change the end amino acid. It will be different. And so our mutation is going to be as such, a CCT. So this time we've changed the first position, CCT. Now when we make the RNA from this, you'll see we have a 5' to 3', and now we're going to have a GGA. And let's ask what kind of amino acid we're going to make from that. Let's put our code back here. And we're going to need a GGA. So what's a GGA? Now we've made a glycine. Now this is a pretty good substitution here. We've gone from a positively charged amino acid to a non-charged amino acid here in glycine. This is very likely to lead to some kind of change in the actual protein. Not only did we bring in a new amino acid, but we brought in one that is structurally quite different than arginine. Now, for the exam and anything in genetics, you're not going to need to know that arginine has a positive and glycine doesn't. That'll come later when you take biochemistry or maybe cell biology. You'll cover that. I erase that for a little bit because I want to talk about some features of missense mutations. First is that they always change amino acid sequences. Usually change protein function. So there are two options here when you think about a missense mutation. It may change protein a little, because you're still forming the same protein except one amino acid has been changed. So often it's not a huge change, it's just a small change. However, sometimes you can change it a lot. A case like sickle cell anemia, that's just one amino acid change, one nucleotide that's been changed. So sometimes it can be a big change and sometimes it's a little change. Now I want to talk about a nonsense mutation. And we're going to use the same DNA here, and this time we're going to change it a little differently. Again, we're still just going to change one nucleotide, and this time we're going to change the first one. And we're going to make this an ACT. Again, we're changing that first one. When it's transcribed, we're going to have a 5' to 3'. A goes to a U, then G, and then an A. And if you look at our genetic code, right here, you'll see that a UGA is right here. It's a stop codon. And that's what nonsense mutations are. Nonsense mutations change an amino acid, I'm going to abbreviate that as AA, an amino acid to a stop. Now the question is, does that change the function of the protein? It usually does. Let's go ahead and erase this. I'm going to draw something. So it depends on where it occurs. So if you have a mutation, and let's say this is our gene here, our coding strand, or I mean our template strand, and it makes our RNA, which then makes our protein. Now if we change one of the late codons to a stop, let's say our stop is supposed to be here, and we change it to one right before it. Well, what we've done is we've still made the same RNA, but now we have a premature stop codon, one codon over. Often when that happens, you're still going to make a protein very similar to what you see here. It's less likely to have an effect. However, if that mutation occurred very early on, say right here, and you changed an amino acid coding codon to a stop codon, now the RNA itself might not change much, because that's going to still probably have the terminating sequence somewhere down here, and so it's going to still be about the same RNA. But instead of having the stop codon here, we now have the stop codon here. And that's going to change the protein drastically, because instead of reading the whole messenger RNA to make the protein, now we're just reading a very small part of it. And you're just going to have a little bit of that protein I'm trying to signify here. So nonsense mutations usually will cause an effect. Depending how close they are from their predicted stop codon, where they're supposed to stop, will often factor into how serious of a mutation that is. A mutation down here, not as serious as a mutation down here, up here. And this next one I'm going to talk about is called a neutral mutation. Since I told you you don't need to memorize the different charges of the amino acids, I'm going to just explain in words what a neutral mutation is. It's when you change the sequence by just changing one base pair, typically, and let's say originally it charged for a polar amino acid, like arginine, the polar amino acid, and now the substitution has changed to a different polar amino acid. It could be from an arginine to a lysine. Again, you don't need to know arginine and lysine are both polar molecules, but that's what I'm talking about here. It could also be from a nonpolar amino acid to a different non-polar amino acid. And we say these are neutral because often what happens here is that the change is minimal. It does change the amino acid, so it is a missense mutation, but the protein that's produced usually isn't affected because you haven't changed the amino acids drastically. So sometimes these missense mutations can be neutral. Different protein, different amino acids, but the net gain or loss is zero. And those were all base substitutions. Now I'm going to talk about insertions and deletions. Again, I'm not talking about chromosomal deletions, just talking about removing a single nucleotide. Insertions and deletions will cause what's known as a frame shift. Now I should make one caveat here. If the insertion or deletion, if insertion and or deletion is not in a multiple of 3. Alright, now I'm going to draw the messenger RNA here. Remember though, the mutations don't occur on the messenger RNA. The mutations occur on the DNA, but what we see, but the effect of that mutation is more easily seen on the RNA. And so let's say this RNA is broken up into a series of codons. And we'll just start with our start one, A, U, G. And then we'll go A, U, U, G, U, G, C, G, A, C, U, and then we'll stop here with a stop codon of U, A, A. Now this is normally going to make a protein that is methionine, isoleucine, cysteine, alanine, threonine, and then our stop codon. Think about what a single nucleotide deletion might do. So we're just going to remove one of these nucleotides. We'll remove that one. And remember, the mutation occurred on the DNA, but we're just showing it here on the RNA. Now that has changed our reading frame. And so that's going to change everything. So how's that going to change our protein? Well, let's go ahead and draw that here. We're still going to have AUG, so we're still going to make methionine. However, we have to read this differently now. Now we have to read AAU, and AAU is asparagine. And then the next one is going to be GUG, and that's going to be valine. And then CGA is arginine, and then ACU is threonine, then CUU is leucine. And then see, you can see here what happened now is we've lost the stop codon. And now I didn't have room to draw, but remember this messenger RNA is much larger than what you see here. So it's going to keep reading this out of frame and keep adding different amino acids here until it randomly and by accident comes across another stop codon. So this will typically either lead to a premature stop codon in some cases, or it might lead to a much longer protein made. A later stop codon is formed. But the most important thing you can see here is that these amino acids have changed. Isoleucine now is asparagine, cysteine is valine, alanine is arginine, threonine. I did this completely by random, but it just so happens to be that this particular amino acid didn't change because of the degeneracy of the genetic code. Now, but importantly here, instead of a stop codon, we now have leucine. And again, any other amino acids that follow. Now, we're not going to talk about it or explain it, but you can do it on your own. You could also imagine how if we inserted a nucleotide in between one of these spaces here, say a G, we place that there. That's also going to change the reading frame and change the actual protein that is made. So these are frame shift mutations because they change the reading frame of the gene. So let's draw the DNA, and I'm going to draw using the template strand, so 3' to 5'. This is our DNA. And then our RNA, messenger RNA, is going to be 5' to 3'. And mutations can occur at places outside of the reading frame. So we may have our reading frame right here, starting with our AUG site in the messenger RNA for starting and then stopping. But you can have a mutation in other places that can also affect the actual protein that's being made. It can be in the untranslated region, so the 5' UTR, or the 3' untranslated region. That's going to affect the longevity of the messenger RNA that's ultimately produced, and maybe the ability of it to bind the the ribosomes during translation. We could also have mutations in the introns, in the 5' splice, the 3' splice, or the branch point. So we can have mutations there as well that can affect the actual protein being made. You can have mutations in regulatory sequences, either the promoter, enhancer sequences, or silencer sequences. So this should be a bit of a review for you, but as we're thinking about mutations, it's a good idea to kind of keep these in mind as we move forward. And let's bring up some terms here. Often we use the term wild type. And when we use the term wild type, we mean it's a non-mutated gene. It doesn't mean we got it from the wild outside of the laboratory. It means it's the non-mutated gene. Then we can have different alleles of this wild type gene because of mutations. And so wild types, after they get mutated, are called mutant alleles. So mutant allele is just a copy of the wild type gene that has one or several mutations in it. These mutant alleles can be harmful to the organism. They can even be lethal, in which case we would call them a lethal allele. They can be neutral, meaning having no effect at all, which most of them turn out to be. Or they can be beneficial. And then kind of some of these specialized mutations. One could be a conditional. And this last one could be a suppressor. I'm going to spend some time talking about these last two. The first ones I think are more straightforward. A mutation that is harmful means that it doesn't protect the organism as well. A neutral one can be one of those mutations that doesn't change the amino acid, so it doesn't have much of an effect on the organism. And beneficial mutations are ones that make that protein perform stronger or better in certain environments. But let's talk about conditional and suppressor mutations first. That is in a little bit more detail. The best way to explain a conditional mutation, so let's talk about that first. Is that at one temperature it behaves one way, at another temperature it behaves another way. And one of the classic examples is a fly that can fly versus a fly that can't. So let's just draw a graph here. And over here we're going to put wing development. And here we're going to go temperature. And we'll go from 15 degrees Celsius all the way to 37 degrees Celsius. And at a lower temperature in this particular mutant, well, first of all, let's draw wild type. Wild type is going to look like this, if this is 100%. Between 15 and 37 degrees Celsius, the wild type will form a wing. However, when a mutation has occurred, a mutant allele will be like this. At a low temperature, it'll form a wing, and as the temperature decreases, or rather, increases, wing development goes down and you have no wings. So over here we have wings, and over here's no wings. So when the embryo is developed at 15 degrees Celsius, you form wings. At a higher temperature, as the embryo develops, no wings. Why is this? Well, that's because at different temperatures, again, remember, when we have the mutation, it changes the protein. And even if it's just a single amino acid that's changed, sometimes at different temperatures, that protein can fold differently. It's still going to fold, but just differently. So maybe at 15 degrees, the protein's going to fold something like this, where we have this nice active site formed here. Maybe at 37 degrees Celsius, that active site is no longer accessible. And maybe that active site in this scenario is important in binding certain components in order to have wings develop. Okay, so again, at low temperatures, conditional mutants have one phenotype. When you raise the temperature, they have a different phenotype. And it's because of the way the protein folds at these different temperatures. Again, it can just be one amino acid change, but it can have a different effect at different temperatures. Not all mutations are conditional. Very special and specific ones are. And I want to talk about suppressor mutations. And so what I want to do with this is first draw, let's say this is our wild type here. And then we have a mutation. We'll call this our mutation, or our mutant strain here, or gene. And there's just a simple mutation on this. But that mutation causes the protein to fold, and I'm going to use just very generic geometrical shapes here. And let's say it makes it fold like a triangle. No protein folds like a triangle, but it's just an analogy. The wild type one I didn't say before, but it folds like a circle. Sometimes you can get a suppressor mutation on that same gene. Here's the first one. And let's say we have a new mutation, and let's put it here. And that allows this suppressor mutant here to now fold differently. And in fact, it'll fold just like it does at wild type. So it suppresses this original mutation. And more specifically, we will call this an intragenic. Intragenic, because it's a mutation that occurs in the same gene. Now let's draw a different mutation. Again, we have our original mutant here that instead of forming a circle protein, it forms this triangle protein. And then we can get a mutation that doesn't affect this gene, so we still have this mutation here. But in a different gene, we get a mutation in it. And somehow this mutation in this other gene affects the ability of this one to form a triangle versus a circle. And because of that, it will now form a circle. Because the mutation is in a different gene and not in the original gene, we call this an intergenic suppressor mutation. Again, intragenic suppressors are mutations within the same gene that was originally mutated. Intergenic suppressor mutants have mutations in different genes than the gene originally mutated. This one's still here, that mutation's still there, but this somehow or another compensates so that we're able to, once again, form the correctly folded protein. And the last thing I want to talk about in this podcast is to talk about the difference between a somatic mutation and a germline mutation. Remember, somatic cells are all of the cells in our body except germline cells. And germline cells are those cells that will make sperm and egg. So somatic mutations occur in somatic cells. They will affect that individual. And specific tissues. If I get a mutation in my liver, it's not going to affect my pancreas. So if you have a body here, right, and they get a mutation in their heart here, those mutations will only affect that region. Another important thing about somatic mutations is that they are not heritable. Germline mutations affect germ cells, those cells that will make gametes. They have a low effect on the individual. If you get a mutation that produces altered sperm, it could lead to something like infertility, but it's not going to cause any health abnormality for that individual. But importantly, they are heritable. And when they're inherited to another organism, the progeny I should say, all the cells in that progeny will have it. So you may end up starting off with this may be the person, and they get a mutation in their germline, and when they pass that on, every single cell in that progeny will have that mutation. So long-term wise, if you think about it from an evolutionary standpoint, germline mutations have a greater effect on progeny and downstream progeny, whereas somatic mutations don't. They're more of the here and now mutations. They affect the individual, but they're not going to be passed on. So to summarize what we learned here, you should know that point mutations change the nucleotide. They can either be a base substitution or they could be an insertion or a deletion. The effect on the protein can be varied from silent. We talked about silent mutations, we talked about neutral mutations, we talked about missense, we talked about nonsense, we talked about frame shift mutations, we talked about mutations outside of the open reading frame. We talked about different kinds of mutants, mutant alleles. We have mutant alleles that can be lethal to beneficial. We spent a little bit more time talking about the differences between like a conditional mutant and a suppressor mutant, and the two types of suppressor mutants, intergenic and intragenic. And then we close with the difference between a somatic and germline mutation. Alright, that's all we have for this podcast. If you have any questions at all, please let me know. See you in class. Bye.